Right. Hi, everyone. We are the Nonsense Mediated Decay team. I'm Federico, and I will present the first half of the talk. As we have heard, this Dream Challenge was all about hypercholesterolemia, which is a complex trait influenced by several factors, from diet to genetics to lifestyle. And the disease is characterized by high concentrations of LDL particles in the blood, low density lipoproteins, which lead to the formation of, yes, okay, sorry. High, so it's high levels of LDL particles, low density lipoproteins, which form these fatty plaques in the blood vessels. And these in turn lead to a number of cardiovascular issues such as artery disease and stroke. So in the PEGS cohort, up to 33% of the samples were diagnosed with hypercholesterolemia. So it's a disease that shouldn't be taken lightly. And as we've heard, the first task was disease classification. So we had to build a machine learning model that could classify people into healthy or disease using the PEGS data. So just a summary of the available data. There were in total more than 9,000 samples, which were evenly split into training, validation, and test sets. And we were allowed to train our model only on the training set. And although the PEGS data is very multidimensional and multimodal, not all of the modalities are available for all of the samples. For example, the health and exposure survey data and the geolocation data are available for all of the samples, but the exposome survey is available only for about a third of the sample. And the genetic data, so whole genome sequencing and methylation, are available for only about half of the people. So for our first submission, we focused exclusively on the health and exposure survey because it was available for all of the samples, and it contains almost 500 questions ranging from family history, disease history in the family, which medications you're taking, which diseases you have, and what is your overall mood. So we reason that this could be potentially correlated with hypercholesterolemia because hypercholesterolemia leads to cardiovascular issues, which can potentially be correlated with other with the overall health status of people. So we did a very simple pre-processing pipeline. We simply removed all of the variables that were not informative, such as the date when the questionnaire was completed. And we also removed the free text answers because they had just too much variability. And the remaining variables were just converted to the appropriate type, so either categorical or numerical. And if a variable had too many missing values, it was ignored. Otherwise, the missing values were imputed with the median of the sample. So having cleaned the data, we decided to apply a random forest model. And we chose random forest because it achieves a good trade-off between interpretability and predictive performance. Another reason we chose random forest is that it can automatically do some feature selection and feature engineering. So, for example, if the questionnaire has a question like how many cigarettes do you smoke in a day, random forest is able to manipulate this variable, and if necessary, it can convert this variable into a Boolean variable if that increases the predictive performance. We submitted this simple random forest model, and one of the best part of this challenge was that there was this leaderboard where you could compete with other teams. And although this model was very simple, it achieved quite good predictive performance with an area under the curve of 0.758. And at the time, it was already the second top model in the leaderboard. Yes, so then we focused, so we reasoned, okay, we are onto something here, but how can we improve the model? And we decided to focus on the genetic components of hypercholesterolemia. because from the literature, it is known that there is a condition which is familial hypercholesterolemia, which is inheritable, and it's caused by mutations in genes that are involved in the cholesterol pathway. So if you have a mutation in, for example, the receptor of LDL particles, it's easy to see why you have hypercholesterolemia. But on the other hand, there are people who don't have any obvious mutations but still have the disease. And if you look at the genetic landscape, as is often the case for complex traits, there are many variants that have a small effect size. And perhaps taken individually, they're not significantly associated with the disease. But if you have a particular combination of alleles, then you are more likely to have the disease. And to exploit this, we leveraged polygenic risk scores. And just a few words about how they are trained. They are obtained by doing a genetic association study, which is a regression that finds the effect of each genetic variant on the phenotype. And then when you want to score an individual, the score is simply a linear combination of the alleles contained by that individual, each weighted by the effect size of the allele. And these polygenic scores capture both the mutations in the genes in the pathway of cholesterol, as well as the polygenic component of the disease. And it has been shown that people who do have the disease, colored in red, do have higher polygenic scores. So we went to the polygenic score catalog, which is a repository of scores, and we found already 12 sets of weights for hypercholesterolemia. We downloaded all of them, and we applied all of them to the individuals in the PEGS cohort. But now there is a problem, which is how do we integrate the random forest prediction with the polygenic score prediction? Because these two models, these two features are calculated independently so far, and furthermore, not all of the samples have genetic data. So the samples with genetic data and with polygenic scores are only a subset of the total samples. So we decided to do as follows. For the individuals that do not have genetic data available, we cannot improve the random forest model. So we just use the random forest prediction as the final disease probability that we submit to the platform. But for the individuals that do have the genetic data, we build an additional data set that includes both the random forest prediction of the previous model, as well as all of the polygenic risk scores. And on this new data set, we train a logistic regression, which we then use, then we use the output of this logistic regression as the prediction for the individuals with genetic data available. And this is a way to, in a sense, regularize the random forest prediction and complement it with the genetic data. And at the same time, we don't have to choose one polygenic score, but we can use all of them at the same time and find the best combination. Yes, so for this model, we found that the predictive performance was much better. We went from an AUC of 0.758 up to 0.775. And this was one of the top models in the leaderboard. So these are all of the models that we submitted, 6 in total, although the model that I described is just the best that we could achieve, the top performance that we could achieve. But we did try other possibilities, and I will briefly talk about a few of them. So the first thing that we tried was to use open targets data to also, again, exploit the genetic data. And open targets is a platform that integrates publicly available data to find genes associated with the disease. So our idea was to train a linear model on the genetic variants that fall in the coding regions of the open target genes for high cholesterol. And you see here, there are already quite a few genes that are associated to hypercholesterolemia in open targets. But unfortunately, this led to a worse predictive performance. And we also tried to incorporate the exposome surveys data, which are questions about lifestyle and exposure to chemicals. We tried to use a similar random forest scheme, but this also didn't improve the performance. So in conclusion, we found that simple models worked better than more complex models that we tried. And for this challenge, it was very useful to leverage pre-trained data. I'm talking about the polygenic scores, which were trained on a much larger sample size, and we were just able to apply them to improve our prediction because the sample size in the PEX data is not huge. And at the same time, we a bit regret that we weren't able to leverage more modalities in the PEX data. But this means that there is a huge potential to improve the estimate because our model was very simple, but it performed well. And in particular, with the PEX data, there is a huge potential to find gene by environment interactions. And this was the topic of the second part of the challenge, which Alessandro will talk about.